Welcome back, everyone. Thanks for joining us for session 2. I'm hoping that many of you were part of session 1 and the enthusiasm is there for what we will do in session 2. Our session 2 is about future use of population descriptors in genomics research. We touched on some of that earlier this morning, and we're going to hear from some excellent speakers who are going to follow up on that conversation. This session will be moderated by our committee member, Dr. Rick Kittles, who is professor and founding director of the Division of Health Equities in the Department of Population Sciences at City of Hope. He's also the associate director of Health Equities of the City of Hope Comprehensive Cancer Center. You're on, Rick.
All right, thank you, Charmaine. And thank you to our speakers who are joining us now for this second session of today's workshop. And we're building on the first session, which took a look back in the history of race and racial categories. And now we will be discussing how population descriptors may be useful in genetics research in the future. So we will discuss the diverse types of population and individual descriptors, and possible ideal descriptors of populations and individuals for future genomics research. Again, we'll have about 15-minute presentations from each of our speakers, followed by a discussion with the committee. And I'll again remind the audience viewing the webinar to please submit your questions in the box on the event webpage. So our session today will begin with a talk by Dr. Tess Mersha of the University of Cincinnati, followed by Melinda Mills of the University of Oxford, Joanna Mountain, who is recently retired from 23andMe. Then following that, Emir Kenny from Mount Sinai. And finally, our last speaker will be Malia Fullerton of the University of Washington. So again, for the sake of time, I won't be reading their full biographies, but you can find them in the briefing book on the event webpage. So with that, I'm going to turn it over to our first speaker, Dr. Tess Mersha, who is the Associate Professor of Human Quantitative Genetics in the Department of Pediatrics at the University of Cincinnati Children's Hospital Medical Center. Tess?
Thank you so much, Rick, for the invitation, for the committee, and for the interaction. So let me put in I hope you see my slide. So I'm going to tell you a little bit of connection of the topic and my own research, but the title of my talk I put like here is the race, ethnicity, and ancestry in genomic research. I'm going to tell you some of the best use, the misuse of these terminologies in genomic research. So the outline of my talk follows like this. So I will discuss little bit of race, ethnicity in the era of genomics and ancestry, some of the misuse of population descriptor, basically this terminology, and the best practice of moving forward, taking some case studies from our research, particularly using some causal inference approach and some suggestions toward the end. So if you look at the history of genetics, starting to Mendel's basically pea plant, so it has been evolved over the years, and from single qualitative and Mendelian genetics, just pea based on colors, population genetics, quantitative genetics, and basically it's moved to different levels, somehow supported by the technology and somehow supported by the methodological advancements. So this is really great thing coming, because from gene discovery now we're talking about functional notation and causality. So these are moving target. Maybe in the coming years it may go to next level. Whereas, as we discussed in the previous session, the history of human racial classification. So I'm not going to go through all this, how it's classified. Basically, in some cases, it was classified by colors, in some cases by skull size, and in some cases by demography. So you can see how it evolves from from early days to now the U.S. Census in 2020. And now the idea is whether we need to move to Ancestry, leaving aside all these different categories. And there are millions of people in the U.S. They change their racial ethnic identity from census tract to census tract. So, but on the other hand, the concept of race and ethnicity really remains the same. So wherever we can classify, we can perceive ourselves in a different way, but the society perceives us in a different way because primarily, in my view, color is the big marker of this human classification. So using that, if you look at the world global view of race ethnicity, and this is, the US classification system, as you can see, race and ethnicity now in the US Census 2020, there are more details of each groups, like previous speaker mentioned, Middle Eastern type and other groups, and it's more detailed classification. But if you look at the Brazil population, they have white, brown, black, yellow, indigenous. And if you move to South Africa, Afrikaner colors and blacks. So I'm not mentioning all this difference. Even in tiny Cuba, I have different classification than what we see in other parts of the world. So the bottom line is, how do you really harmonize all these different things if you want to really have a unified genomic research across the different worlds? If you want to replicate studies, which population is in order to replicate, or are you just colorblind? So that's a kind of topic I feel that's very important. The bottom line in the slide is, we cannot use the US category to outside the US, as you show in this figure. There are differences in many ways, and self-reported race ethnicity is depends where you talk about it, what's the cultural norm and all those things. Being from Ethiopian origin, my understanding before I moved to the US about race ethnicity is completely different now than a few years back. So it's just really contextual and cultural or location dependent. So what about ancestry? We sometimes feel talking ancestry is a good thing, but in my view, ancestry is also an inferred term. It's a proportion, and what we measure is basically admixture. We don't really directly measure ancestry, so it's a latent variable, and we don't know directly, and because we don't know all the founders of the generation that we have. So it's basically an error-contaminated terminology. So what we measure is admixture. Ancestry is really in an error-contaminated admixture. So it's not a direct measure, and it has its own error. The errors could be coming from measurement, some kinds incomplete coverage of the genome in some ways, missing data, and imperfect founder population. For example, for most of us, I'm always puzzled when we use a Yoruba population for the entire billion population in Africa, so reference population. So you can imagine how the eastern part of Africa should use the West African as a reference population in order to determine some of this admixture and other things. So there is an imperfectity in estimating ancestry as well. So a good example is here how complex race, ethnicity, and ancestry are. So, for example, here is a story. Mr. X, this person was born in Japan, so he considered a Japanese in terms of ancestry. But the person was adopted to Italian family. So he grew up in Italy. So his ethnicity is Italian. And then he moved to the US. So he has to adapt the US classification. Then he's Asia. So you can see how confounded these terminologies are, especially if you talk about genetic research, where we want to see, a one-to-one correspondence type of analysis. So I think this has been already discussed before, but what I want to say is, It depends what we see. If you see in one box, you may see black or white. But if you see the whole continuum of the global population, it's a normal distribution. It's not possible to slice and die in a certain group. So it's more of a gradient. It's a client. So we need to take into account, because the reason being, we don't have enough time to have isolation, and so that it's not a truly biological race that we talk about. It's just one race. There's no human race. It's just there's one human race. So, and again, we cannot prove in any scientific way. If you take a blood from a random person, it cannot assign any racial group. So there's nothing to classify that way. So I said the good and the bad things, but what are the conditions we should use really race? In my view, we need to still use race, because it has its own historical context. Because if you want to refer racial disparity, that's how societies are structured. And if there is unequal treatment and healthcare access case and race-specific environmental condition, then we need to consider race as a factor. And most importantly, in genomic research, if you want to represent all the population, we need to level population by race so that we can access the genetic material, so that we can reduce the racial gap in genomic research. So there are the conditions I feel we need to use race information. And there's also some condition I feel using race is a misinformation in genetic studies. For example, if you are comparing race for evaluating genetic cause of disease without accounting social environmental risk factors, I'll give you an example on that. So this may convey, racial disparities just because of a genetic variant, rather than some of the underlying perhaps structural racism. On the other hand, sometimes, you may see people are using race ethnicity as a covariate. Are they using to explain genetic variation or social, some of the structural racism case or socio-environmental determinants? So it depends what is the context using as a covariate. And also, in some cases, we think that, racial health in disparities we are using race and genetic as essential factors for health disparities. In that case, I think we need to avoid those kind of labeling as essentialism of either genetic or race, but both are really contributing to what we see the disparities that we have now. So the other misuse of racial category, I'm not going through all these details, but is a clinical algorithm. In some cases, we use clinical algorithm to decide to have variations. In some cases, for this one, for example, in cardiology, if the add 3 points to the risk score, if the patient is identified as non-Black, so this gives a guarantee that there is some biology differences instead of the things that we talk about, socio-environmental factors. So there are a number of diseases that are still use racial category in clinical algorithm, and there are factors that we need to use. What's puzzling for me is sometimes pain management. You have to have, if a Black person has 10, it's the same as 8 in the scale for white. So those are things I feel there's no biological base for that kind of clinical algorithm.
3 minutes, Tess. You have 3 minutes.
Okay, I'm almost done. Thank you. So a good example again here is that, in some cases, for good intention, people do nice research here to the racial disparities and preterm births, but some news media outlets may take in the wrong way, saying study points genetic disparities in preterm birth. So these are really, we need to address this issue before it goes far labeling genetics as the real cause of racial disparities. Again, ancestry here, it shows that, the higher the African ancestry, the lower the income. So this is again a misnomer in that case. This African ancestry could be a surrogate for less education, fewer parental schooling, inheritance, less wealth. So those factors should be considered before just associating African ancestry with with low income or some of these risk factors. And this is another example which shows African American race associated with AD, atopic dermatitis, independent of African genetic ancestry. In this case, race is associated to AD, but not ancestry. This could be, a reason why socio-environmental factors could contribute to these differences. So I think in all studies, what I want to show this slide is that we need to have a good adjustment of for studies that we're intended to do. In this case, this is a type 2 diabetes study where, for Mexicans, before adjusting, you find high p-value, also for Colombians, but after adjusting for socio-environmental factors, in some cases, you lose all those significance, and in some cases, it attenuates. So there is an important thing. We need to adjust our all studies as much as possible. So this figure is showing you again. We always say there is differences based on education, but same education level, college graduate level, you see. Pregnancy-related mortality ratio in blacks higher in blacks than in white, 5 times here. So could this be, being all college graduate, could this be, due to structural racism or some of the other things? So we need to consider all those factors. So the case study I mentioned is to use structural equation modeling. Andy mentioned, direct association of genetics to a disease, and in some cases we directly associate biology to disease. But I want to implement here in terms of structural equation how we can really disassociate the relationship. So the first thing, risk to asthma in my study, risk to socio-environmental factors and asthma. Now we're going to use ancestry rather. So this is a traditional study from genetics to asthma, but when to use the mediation analysis where mediators are socio-environmental factors, and that's what we got in this study. I'm sorry, this figure is a little bit overlapped, but the idea is if you look ancestry in the middle of this figure, there is no significant association to asthma readmission. What is associated is race, ancestry was associated to hardship, disease management, those are associated to outcome. So I think if you dissect this relationship, we will find the real mechanism rather than just one-to-one ancestry to readmission. So in concluding, race should not be used as a proxy for biological risk factors. Race is a proxy for social structure and racism, and in some cases, emphasizing on ancestry could lead further disparities. And this disparities racial gap is actually confounded by inequalities in genomic research. And the point is how we really standardize in this measures of race ethnicity. So here's my recommendation. The first thing is, I think we need to develop a worldwide acceptable population descriptor like ontologies, like we know the genome ontology, phenotype ontology, and especially for large-scale worldwide consortia, so that we have a common language across the various world. As discussed before, I think we need also a descriptor about socio-environmental and cultural determinants. So we need to harmonize all these factors in order to collect and define all important factors. And finally, we need to consider the use of race, reconsider the use of race correction in clinical algorithm. Thank you very much. That's it.
All right. Thank you, Tess, for that very thought-provoking talk. Our next speaker is Dr. Melinda Mills, who is the director of the Leuvenherm Center for Demographic Sciences at the Newfield, and also Newfield Professor of Sociology at the University of Oxford. Melinda?
Okay, thank you, Rick. I'll just share my screen here. Okay. There, assuming everyone can see it. So yeah, thank you very much. And I think my presentation leads on very nicely from Tez's as well. So I just wanted to tell you first a little bit about a study I did with Charlie Rahal, also shown here, where we decided to reflect a few years ago And we engaged in an in-depth review of scientometric review of GWAS studies. So we looked at the authors, we looked at who funded them, we looked at the diseases, we looked at ancestry, we looked at where the studies took place, who was in the studies, because we started to notice when, engaging in GWAS studies ourselves, that it seemed to be quite selective. And coming from a social science background, this was really striking. This is something that we usually try to avoid. So I think many people know this, and we also found the same thing within our study, that around at that time, 80 to 90% of the discoveries were of European ancestry. So this just shows a bubble graph showing in the discovery and the replication samples that, of course, as we saw, many of them have been from European ancestry, and these are the common categories. It was discussed in the last session that we didn't invent, but these are the ones that are often used within the literature and within the statistical methods as well, too. But what we noticed as well, when non-European samples were included, it was particularly for replication and not for discovery at that time. So, and I think it comes back to, how do you even define this? And the last session talked about this in detail. And I think, coming back from this as a social scientist, we just can't reiterate enough. And we've written about this, about it's race and ethnicity is a socially constructed biological category. This is different from ancestry in genetics and genomics. And I'm sure some of you have seen this picture, of how some people even reify these ancestry categories themselves. This just shows someone with a tattoo. And as we know, when the reference panel changes, these numbers can change as well. So probably not a great idea to get that tattoo. But also, I just published something a couple of days ago with Ian Matheson, sort of reflecting on these, on population stratification and population structure. So we have these different ancestry groups, and we know that all human populations, that they have some degree of structure, and the most obvious one is the geographic structure. We've talked about that before. And then due to non-random mating, it's often based on geography, so where you're located. Some variants can be more common in one part of the country than the other, or one region of the world. And so these phenotypes can also, not just genotypes, but phenotypes can also vary geographically in a very non-random way. So that's, these different ways of organizing and thinking about the world, I think, can often become very confusing for people. So back to our 2019 study, we wanted to look at the top terms that were used in these GWAS studies by geneticists in their genetic discoveries. So if we remove the European, which was by and large the largest group, and we just look at some of these categorizations, and we use natural language processing to try to pull out some of the main things, we saw that there were these ancestry categories that were quite fluid. Sometimes they were based on nationality. Tez was just talking about that, or geography. Sometimes they had a political element to them. And this is in, genetic studies. And the prominent groupings were often very geographically based. Many people have said this, but they were also very unclear. So we had African-American, we had in some Black British, Indian, Asian, and then Latino or Latin American. And these became very sort of fluid terms that were used by geneticists in their GWAS studies to define their populations. So definitely the reason, to have some sort of categorization, our analysis shows that definitely is required. And what we also found very striking is we had learned a lot about ancestry. There have been several studies such as ours, but the country of recruitment was really striking. So 72% of all GWAS discoveries, up until 2018, when we looked at it, came from just 3 countries, the UK, the US, and Iceland. And everyone, of course, knows about DECODE. And that's quite a small population. And I'm a demographer as well. So if we look at, if we adjust this for population size, this Icelandic population that has about 355,000, represents about 19% of these discoveries, compared to the US population that's about 330 million, represents a smaller amount. So it's really interesting from a sort of science perspective to sort of think about, who we're looking at and how that will influence our results. We also looked at the scientists. I don't have time to cover that. I can talk about it later, or you can look into the article. But we also looked at who funded it and sort of the power structures in genetics and genomics. And we found, you'll see here, you have the funders here, and you can see which sort of ancestry groups they fund, but also how they fund different traits and how some traits have more or less funding in them. And remember that, when you're looking at the UK and the US, and to some extent Iceland, these are specific comorbidities and different interests within these populations. So there's whole other populations and diseases that haven't been looked at in detail. And then we thought it was interesting, given this lack of diversity, we thought, well, we should probably track it to try to have some accountability over time. So we published this in 2020 in Nature Genetics, and it's an online tool, and we update it every morning at 6 a.m. Based on the GWAS catalog to see, well, just how much diversity is there. And rather disappointingly this morning, this is from this morning, we can see that diversity is actually, gone down over time. So you can see here that it's about, 96%. If we use these rough ancestry categories, you can see that it's because of the UK Biobank largely that it's gone down actually over time. So you can look at, in terms of by year, the disease categories that are covered in geography, and as was discussed in the previous session as well, you can look at the participants by ancestry, but if you look at actually the associations where they're discovered, you'll see differences as well too. So in this, on our GWAS diversity monitor, you can real-time search into different phenotypes, and then you can click through on these actual studies. You can see how many have been done in these different populations, and it clicks you through to the actual study. And then within our study, we also wanted to look at what were the, who's being studied and what are the characteristics and the traits, because it's very unusual from a social science perspective. And this has come up very often to have things that are absent of, who's in the study. Is it women, men? Are they older? What country do they come from? And this just shows the top 10 data sets. And we actually had to code that by hand with different people doing it. So that would be another suggestion, definitely for editors, to try to get more structure into which data sets you're using and which part of the sample. And I mean, what we found, just to summarize it, of course, you'll know the European ancestry part, but, there were mostly industrialized countries. We know that there's large data collection projects now in Africa and elsewhere, but these were quite specific populations. They were often older. There was a sex ratio balance more in women, and a lot of them were non-representative samples like the UK Biobank. And this would really, we've seen this, some simulations published recently in the Lancet, showing that this really these GWAS estimates are biased towards the overrepresented group. So if you have a group that's healthier, lower BMI, non-smoker, that will be different in your discovery and your target population. So these are things I think that remain very important beyond ancestry. And we published about 5 years ago in Nature Human Behavior. We did some simulations and also worked with 7 different populations and birth cohorts. And we showed actually that these genetic results differ a lot compared from the country that you come from and the birth cohort. Not really big news to the demographer, but this was something that we saw that was often ignored when we were combining these very heterogeneous data sets together. And there's been other studies as well showing that actually within ancestry groups, you get this huge difference in polygenic score prediction by socioeconomic status and sex and other factors. Oh, there we go. And then I come back to, the thought about what we often look at and within the social sciences. And this has been mentioned by multiple people, it's about cumulative disadvantage and intersectionality. So I guess the question would be, can we really pull race and ethnicity out of this? And, Tez was just talking about this as well. We get this cumulative disadvantage that's related to your socioeconomic status. It's often overlapping and intersectional with your race, your nationality, where you live, your health, your sex, when you're born, and all of these factors play a role. So I think it's becoming increasingly difficult to pull out just one trait, given that there's so much collinearity and overlap between all of these different categories. And just to give you an example before I finish is just looking at this is polygenic scores. So if those are that are in the 90 plus category for the educational attainment polygenic score, so we have people 90 plus, and these ones, and this just shows we were talking a lot about G by E, but it just gives you an example. You know, we have, if you have one college educated parents, you have a substantially higher educational attainment, level of educational attainment, if you're in the exact same polygenic range than those without a college educated parent. So that just shows that the social economic environment and what we were talking about, this context is just so key, and it can really influence polygenic scores, and it's been shown to influence diabetes, cardiovascular disease. So there's a lot more in the polygenic scores than we realize. There's a lot of social structure. So just to conclude, many of these recommendations are from our 2019 article, but, you know, just standing back and looking at race and ethnicity and the intersectionality and the just cumulative inequality of, you know, including and thinking about multiple types of diversity. Also, it's been discussed already, so I can go through that quickly, but just a more careful interpretation of these genetic differences and what actually is, you know, what's contained in the polygenic scores and the genetic discoveries we have. There's a lot of population stratification and population structure in it. I think participant and researcher involvement remains key. So people from different communities being involved. So I'm on the board of Our Future, the scientific board and ethics board of Our Future Health. It's a new 5 million data collection initiative within the UK. We're involving a lot of the community, and we're trying to get more hard to reach communities as well and look at socioeconomic and ethnicity differences. And another issue, what we really saw is it's also in the system of how this all works. And this has been discussed before. So just inequalities and authorships and people from those communities not being involved as main scientists, some helicopter science in the past as well too, not involving in building the infrastructures, and just really thinking about the incentive structures in these studies as well. And I mean, what we saw when we analyzed this data, that we really needed a lot more coordinated governance and guidance. That's why we were so happy to hear about this, because you really need what Tez was talking about. You need this broader international ontology. You need the editors, you need the funders, and you need this unified approach to try to push this forward. But you also should monitor it. So that's why we set up the GWAS Diversity Monitor. It should be expanded along different categories beyond ancestry, but there has to be some consequences for this. So thank you very much. I think I'm at my time now, but those are the kind of the main points that I wanted to discuss now. Thank you, Rick.
Thank you so much, Melinda, for those thoughtful comments. Our next speaker is Dr. Joanna Mountain. She is a consultant at 23andMe. Joanna, you're muted.
Yes. Thank you, Rick. And I'm just going to share my screen here. So I'm Joanna Mountain, and as Rick said, I retired from 23andMe after 14 years as Senior Director of Research. I continue to consult with 23andMe, and I continue to consult. And I wanted to today, think about the last 15 years at 23andMe and how at the company people have used labels, population labels throughout the service and research. A lot has happened in those 15 years in terms of thinking, writing, and research on this topic. So, while at 23andMe, I aim to kind of keep up with the guidance that was coming from people such as speaking here. But let's go. One of the first things that I pushed for early on was to use, rather than the racial and ethnic labels, was to use labels that are closer to geography. And so this is an example of a question that we put to our customers about their origins, and it kind of tries to go below Europe and ask, if they have connections or ancestry from Western, Southern, Eastern, or Northern Europe. And so that's one way we started out. We did find that customers were not very comfortable with this geography way of looking at the world. And that did cause us some issues. So we ended up going into other directions, and we asked about cultural groups, such as those listed here on the left. And people were quite willing to tell us about their cultural affiliations. And more recently, we used the ethnologue to get at languages and language groups. That really helps us kind of interpret any genetic clustering that we see in the data. So that's typically how we do it. We do the genetic clustering, such as described by Andy Clark earlier today. And then we say, well, wait a minute, what is similar for the people in this cluster? And sometimes it's language, sometimes it's cultural group, sometimes it's a geographic region. So it's been kind of fascinating to watch that evolve. Now, the reports, including the ancestry reports, tend to go back to the geography for the most part, and here's my own results, pointing to Northwestern Europe, British and Irish. And it is true that I have grandparents and great-grandparents from the London area and the Dublin area. So those are picked up in my genetics, by comparison with, millions of other individuals. I want to say a little bit, beyond the European, if you look at the major broad categories, they do go towards the kind of quasi-racial descriptors that were discussed earlier. But I'll come back to the Indigenous American breakdown, because that's really something recently done by 23andMe and really important to how we thought about that. I want to now jump to the polygenic risk scores that have been such a topic of conversation today. And I'll, spin that off from my own result on high cholesterol, where my report suggested I had a higher than average chance of developing high cholesterol. And the circle here really tells what's happening here. It says, 72% of females with my genetics like mine, do develop high LDL cholesterol by their 70s. And the key thing here is this is based on data from 23andMe research participants of European descent. So the kind of comparison population is spelled out right here. And why is that? The thing is, I told 23andMe that I was female and that I was of European descent. So that way 23andMe can customize the result based on that information. Now, 23andMe often uses the term ancestry, but interchangeably with ethnicity, it seems. But I want to point out here that in the educational materials for customers, there's quite a bit of discussion of ancestry and ethnicity, pointing out the challenges for polygenic risk scores in terms of ancestry, and that you can't really transfer the scores from one group to another. And the last sentence here, as a result, polygenic risk scores trained with data from individuals of European descent typically perform worse among individuals of other ancestries. So trying to be transparent here about the issues that come up with polygenic risk scores. So I want to get into a bit of detail here around the polygenic risk scores, and this is the cholesterol one again. So the models were trained, initially with individuals of European descent. So that's 617,000 individuals who gave us data on cholesterol. And then the training was done with over 500,000 individuals of European ancestry. However, for the other groups, there was an attempt to train using data from each of the other groups, and then testing was done. But you can see that the sample sizes, the N here is lower for these other groups. And so that's quite challenging. But the nice thing is, going to the next slide, that if we look at this odds ratio for the various groups, even though the sample sizes are lower for other groups, the power to kind of provide information for the customers is, the odds ratios are, close to 2 or higher than 2 for all the groups. And that means that there is power, despite the lower sample sizes, to differentiate between people with high risk for high cholesterol and low risk for high cholesterol. So it really shows that you can, with the sample sizes that are here, you can still have the power to develop polygenic risk scores that are appropriate for people of other ancestries. However, I will say that many of 23andMe customers describe their ancestry or ethnic affiliation as other. And in that case, it's much harder for 23andMe to provide the polygenic risk score. And so they typically just provide the score that would be provided to a European individual. So many challenges that are recognized by the scientists at 23andMe and continue to look for solutions. I want to go back here because there's a link to a white paper here that really goes into the technical details of how these scores are developed. And it's well worth looking at the effort that's made to make sure that the models meet the standards for all populations. And that's typically the aim for the release of any health report at 23andMe. So jumping back down to my final slide here, I wanted to touch on the group labels that 23andMe provides for Indigenous Americans, and you'll see them over on the left, Alaska, Great Lakes and Canadian Northeast, South Central Plains, Southwest, Great Basin, and Columbia River Basin are the Indigenous regions. And those were developed, with guidance from experts, including Indigenous scientists, who really helped, with the development of the content around these results. And the nice thing is that the page that was developed with the educational information is just easily discoverable by customers and is actually viewable by anybody. So 23andMe really thinks that one of the solutions is to provide educational material and context so that people understand, where these labels came from and how things are defined. And I think that is critical. We don't know that everyone reads everything, but if they get a result, they tend to read what's relevant to them. And so we encourage that as much as we can. So I've had put a link to a blog post, which it goes into more detail, but just wanted to give you a sense of how we got to providing those particular labels. I'll wrap up and note that, a few of the things that I've concluded from this experience is that granularity you can get more granular than the continental regions that really end up being those quasi-racial labels. You can certainly get more granular than that in various ways. I do feel strongly that self-reported affiliations with groups are critical. We want to hear from individuals who they think they're affiliated with in terms of ancestry. Community engagement is critical. Also, that makes the grid plan is really hard if you're trying to engage with communities, because then you end up having to define the community in order to do the engagement. So the grid approach is tricky that way. So those are a few of my thoughts on recommendations. And back to the polygenic risk scores, I would say as much as possible, it's important to develop and test with data sets from particular groups. And that is mainly can take years to build up the size of the data set that you need to have the power to develop the risk score. And it's worth putting in that effort to build up those data sets for the more granular groups. And I think I will wrap it up there.
All right, thank you, Joanna. That was very interesting, very, very interesting. Our next speaker is Emer Kenny, who is the founding director of the Institute of Genomic Health and professor of genetics and genomic sciences at Mount Sinai. So without any further ado, the floor is yours, Emer.
Thanks so much, Rick. And thanks to the previous speakers, you're setting me up really nicely. So what I wanted to spend the next 12 minutes or so on is really thinking, building off of some of the ideas about using genetic ancestry IVD that Joanna and Andy actually this morning also spoke about, and thinking about how we are utilizing genomic descriptors in discovery and translational genomics, and how that might change as we go into the future. So here are my disclosures before I start. So I've been really lucky to have been involved in a large consortia thinking about big questions at a global scale in genomics for most of my career. And here are some of the studies that I've been involved with. And these days, actually, I'll just point out there's a couple of consortia like the NHGRI funded PRIME consortia that's really trying to redress the lack of diversity in tools that we're using out of these efforts, like polygenic risk scores and human genome reference program that's really trying to build out reference resources that are much more representative of humans on the planet. But in common across all of these consortia really is that these are a set of consortia that are really focused on diverse populations, happily increasingly diverse over time. And most of the consortia, what they have in common is an upfront sampling, either legacy or as part of the work, and assignation of population descriptors through that process and through different study designs and ascertainment. And interest at looking at differences within and between populations as a means to understand not only genetic factors that impact health outcomes, but also environmental factors, whether or not those environmental factors have been sparsely measured at all, or deeply measured within the study. That's a study design question. But inevitably, when you start to look across populations, the population label itself actually helps you, it tells you something about the non-genetic factors in addition to the genetic factors that are impacting health outcomes. And then the long-term goals of all of these studies are really to bring us toward translating research findings to really understand how genetics impacts in real-world populations. At the same time, we've really, through global investments, a little bit differentiated, so investments in different places in different parts of the world. We're really starting to recruit through health systems into biobanks, and these are becoming engines of translation. And this looks a little bit different from the more cohort-based research, in the sense that health systems-based recruitment tends to be much more population-based. Sometimes it's hospital-based. And that sort of gives you a chance to actually see how findings from cohort-based and epidemiological type study designs actually translate into more real-world settings. And as we sort of have a foot in both of those worlds in terms of research, I think this starts to give us a frame for understanding some of the challenges for understanding genetic risk and understanding at a population level. What is being brought up throughout today is that we know we have biases in representation in our genomic databases in terms of georegions or populations in representing many humans in the planet that are not represented in our genomic research endeavors, or underrepresented in our genomic research endeavors, and therefore some of the questions around how we even think about population descriptions just haven't been tackled yet. Also, we're grappling with the complexity in the terms of our own human history on the planet for hundreds of thousands of years, our complex patterns of migration and diaspora, and the admixture processes that have occurred and their impact on the landscape of genetic variation around the planet. And then, as has come up again often today, the need to integrate genomic information increasingly with other types of information. If you're in a health system, you think about clinical history and family history. If you're in public health, you think about social determinants, lifestyle factors. And all of this information really ought to be jointly considered if we want a fuller understanding of what is impacting health outcomes. Something else we're hearing about today is the impact of diversity on polygenic risk prediction, and the fact that genetic prediction can decay if you go out of the research sample from which you generated that prediction tool and into populations. And it's a function of the divergence between the population that you're trying to use the tool and the population in which you generated the tool. And it's for lots of complex factors that we're still really unearthing, and there's a lot of big debate out there whether we'll ever be able to resolve this question in the absence of sequencing every human on the planet. And then when we think about how genomics comes into health systems, I wanted to talk to you a little bit about the health system that I'm in, which is the Mount Sinai Health System in New York City. We are a very large, my dean would like me to say, one of the very largest health systems in New York City. We are a large continuum of health care that covers the life of about 3 million New Yorkers. We have hospital campuses and ambulatory campuses all over the 5 boroughs of New York. If we think about New York, of course, we know New York is a highly diverse city. With New Yorkers really representing many of the U.S. Census race and ethnicity, all of the U.S. Census race and ethnicity groups, and many either not identifying with them or are ticking multiple boxes, which is a city of 8 million people is hundreds of thousands of people. So my health system made an institutional investment about 14, 15 years ago to build out our own biobank, understanding that the types of findings that were coming out of research may not be easily interpretable for patients in our health system, since they look rather different, as at least back in 2007 when we started this. So we started our own biobank, which today has enrolled over 70,000 participants in our health system. So about 2%, and have generated sequence and array data for almost all of them at this point, and including survey data where we ask questions about personal history and demography and family demography. So that if you look back at least 2 generations in participants in our health system, you start to get a picture like this of New Yorkers, which is that 65% of participants in the biobank had 3 or more grandparents that were born outside mainland US. And given the age of the participants in the biobank, about 55, 60 on average, you're now looking at historical events that are coincident with migration histories that are happening about the beginning of the 20th century. And this is really representing people now living in New York who are representing recently over 160 countries around the world. So in that kind of complexity and nuance, how do you think about applying population descriptors? Well, a health system to some degree has to apply population descriptors in some ways for utility and the kinds of clinical algorithms that we use, but in some means, depending on whether you're sitting in your primary your PCP office or your specialist care office, some of this information is incorporated into how you were treated. So we started to look at this in our health system, and this is comparing data that we collect through the BioMe survey that asks these questions versus data that's collected in a health system, usually by a registrar, either self-administered or somebody does it for you. And this is looking at 1.3 or so million visits over the course of about 7 years for 30,000 participants in the BioMe. And you can see that, maybe not surprisingly, the health system doesn't do a great job of capturing population descriptors well about you. It does not a great job for everybody, but it is particularly not a great job if you are East Asian or Hispanic Latino in our health systems. And of course, this is much more nuanced and complex. And if we start to look at regions, subcontinents of the world where people have recent ancestry, and then the types of population descriptors that we can think about using in a place like New York and in the US, actually, particularly how we think about things in terms of African American, Hispanic, Latino, etc., then you can see that there is actually a lot of nuance and a lot of multi-categorizations that are coming into all of these groups. And particularly when we have groups like other, which in New York is not a small group, then you can see it can really run the gamut completely. And of course, we can start to use genetic ancestry to unlock some of this nuance and scale. And without getting into too many details for time, I will just tell you this is using the type of identity by descent techniques and machine learning tools that we build in order to look at what we call cryptic relatedness or relatedness over the course of the last 5 to 20 or so generations in New York City. And there you can start to use machine learning and over the topology of what that looks like to start to find clusters of individuals who share more cryptic relatedness by chance among each other than they do with other people in the biobank. And then if you start to look at the application of the types of population descriptors that we collect, you'll see that these IBD communities, as we call them, are actually very predictive of specific population labels in many of the cases. And this can be important, for example, when you're starting to want to use population descriptors and labels for things like understanding large effect alleles that are inherited in some populations. And the canonical example of that, of course, are the pathogenic variants in BRCA1 and BRCA2 genes that are some of the most characterized genes we know in medical genetics. And there we see by looking at much more nuance than the sort of typical population descriptors we think about in a health system, and actually digging down a lot more into processes of recent diaspora and migration to New York, you will see that we see a much richer picture of the population-based prevalence of these variants across different population groups, often even within population groups that are usually considered part of one more US race ethnicity group. And understanding this prevalence can be very important. And one of the things that we sort of walked away from this type of work as an understanding is that, yes, genetic ancestry in some instances, and depending how you do it, can actually give you a more nuanced understanding, and sometimes even, especially when you're trying to tie medically important variants to demographic events like founder events, an easier representation of who actually would be at most at risk for harboring a variant like that. However, what was very interesting as well is when we went in and looked at health outcomes in patients who were harboring these variants, we discovered that although the rates of personal history of BRCA-associated cancer were very similar between populations, if you were Ashkenazi Jewish in our health system, then you had twice the chance of having evidence of having clinical screening or testing for these variants, which may be due to a lot of things, but certainly indicates that maybe provider or patient knowledge themselves of risk at a population may be something that's important for individuals to seek out medical care. And so with that, I'm just going to give a few thoughts for the future and maybe try and be a little bit provocative. We've heard a lot about how population labels are liable and nuanced and tied to social and political forces, but remain a very important anchor to understand gene and environmental impact on disease. And I do think that we can start to, and we already do this a lot in research, but in other forms, start to think about how we could integrate genetic ancestry into the picture, both as a biomarker for environmental factors and social determinants, maybe imperfect biomarker, but often in the absence of knowing anything else in many of the studies we run, but it also highly liable, but also in addition to ways of incorporating genomic information and population descriptors into genomic research and medicine. And then just, to really sort of center the discussion a little bit on how we're going to inevitably increase how we bring genomic information in a centered way, in a routine way into clinical care, which is going to make some of these questions about population descriptors certainly come out of the research realm and really come in a lot to the medical and public health realm. I think we do need a better way and a framing to really reconcile individual genetic risk and which you can sort of think about almost as precision medicine, to population-based risk, which you can think about as sort of population genomic health. And I will leave it there for now, and we'll enjoy the discussion later.
Thank you, Emir. Before we get to our final speaker, I want to remind the audience that if you want to submit questions, you can do that in the box on the event webpage. And so our final speaker is Dr. Stephanie Malia Fullerton, who is a professor of bioethics and humanities at the University of Washington School of Medicine. Also adjunct professor in the departments of epidemiology, genome sciences, and medicine at the University of Washington, and an affiliate investigator in the Public Health Sciences Division of the Fred Hutchinson Cancer Research Center. So without any further ado, Dr. Fullerton.
All right, thank you so much, Rick. And I'm just pulling up my slides here. Make sure they are in the right orientation for everybody. Let me switch this around. All right, thank you so much again. I really appreciate the invitation from the National Academies and this committee to present to you today with some reflections on the use of population descriptors in genomic research. Before I begin, I would just like to acknowledge with gratitude and humility that I'm speaking from stolen land. The traditional homelands of the Duwamish, Muckleshoot, Tulalip, and Suquamish tribes, past and present. I honor with gratitude that land and the First Peoples of Seattle, and also acknowledge that while land acknowledgement is necessary, it is, of course, not sufficient. So in my short time today, and as I may well run out of time because I haven't practiced this presentation, I'd like to give you my take-homes right at the outset. First, I'm going to try to argue, but I will look forward to further discussion in the Q&A, that clinical translation and the kinds of clinical translational problems that Emer was just describing in her presentation will likely complicate, if not directly stymie, any genes only or gene primary approach to think about population description in the context of genomic research. Slippages and contradictory categories abound in our research practices, and moving beyond these sociocultural descriptors of race and ethnicity may require something more than adopting a scientific genes first or gene only taxonomy or ontology, as I've heard several of our prior speakers both this morning and in our session today arguing for. So that's what I want to argue. These thoughts are informed both by my joint training in human population genetics and bioethics, as well as my involvement as an ethical, legal, and social implications or ELSI investigator embedded in a number of primary research programs and or coordinating centers of several large research consortia, including the Clinical Sequencing Evidence Generating Research Network, or CSER, the Transomic Precision Medicine Consortium, or TOPMed, the Electronic Medical Records and Genomics Research Initiative, or eMERGE, and then the newest research consortium that I've just begun to become involved in, the Polygenic Risk Methods in Diverse Populations, or PRIMED Consortium. Each of these national and international research consortia aim to advance clinical translational research and ultimately precision medicine. The newest, eMERGE 4 and PRIMED, are specifically focused on the validation and implementation of polygenic risk scores in diverse populations, which we've heard a fair amount about today. I also would be remiss not to note that for the past 3 years, I've had the great good pleasure of serving as a co-investigator with an ELSI-funded R01, the Ethics of Inclusion project, led by my colleagues Sandra Lee and Janet Shim, which has taken a close look at the ways that diversity and inclusion are conceptualized in several of these research groups, as well as others that I won't talk about today. So I am to a degree steeped in the current use of population descriptors in this space. And that is the kind of vantage point that I'm going to be coming to you with regard to my comments today. So as clinical translational research studies, each of these research programs is tasked with discovering relationships between gene variants and disease risk as they arise in human populations. The earliest phases of translation focus on basic science discovery, but as the ultimate aim of these exercises, as Emer was just referring to, is the generation of information that can help us to diagnose or prevent disease, research has turned quickly to the development, validation, and implementation of tests that can inform clinical decision-making. And it's there that where, to my mind, the challenges with regard to population description begin. Because even though research participants come into clinical research, bringing with them, as Melinda beautifully demonstrated in her presentation, myriad demographic characteristics and social identities, as their DNA and their data are disembodied and digitized, that complexity, almost of necessity, in order to do the work that we need to do in the context of genetic association studies, such as those that Andy Clark described this morning, much of that complexity is stripped away. So genetic similarities and differences amongst participants can be mathematically parsed as principal components. We've seen a number of depictions of this kind over the course of the presentations today, and these principal components can be interpreted with respect to reference populations of known or presumed provenance, as shown in this analysis of the UK Biobank data with respect to reference samples from the 1,000 Genomes Project. And these principal components of genetic variation can be used to make inferences about genetic ancestry that can help to protect us against those spurious associations, confounding that might be due to cryptic and unrecognized population stratification. But there are many difficulties with this approach, and some of which have been discussed in multiple presentations today, including the frequent recourse to continental ancestral categories, which look and often behave suspiciously like racial biological categories. And this is discussed in more detail and quite effectively and critically in a forthcoming commentary led by Anna Lewis and her colleagues and others who are, I'm sure, are part of the committee, and also have probably watching right now, people who've been participating in a discussion on this issue in the Harvard Safra Center for Ethics. So look for that coming soon. But even though we can do this, and even though there are difficulties with this approach, the more pressing problem, the real big problem here is what happens. And I think we began to get a taste of this in Emer's discussion, and the discussion of nuance. The more pressing problem is when we go from these sort of neatly quantified measures of genetic ancestral relationship, and we try to take the knowledge gleaned by parsing populations in this way, and we bring it back into the real world. So this is a kind of a cartoon depiction. I've been using this for so many years, I don't even remember where I found it, but this sort of notion that when the doctor is encountering a patient in the clinic, that they are going to be able to make sense of information about genetic risk and its relationship to disease incidence that's inevitably being generated, parsed by, understood in the context of ancestry when patients do not walk into the clinic with their ancestry neatly painted on their body, as in the ridiculousness of this cartoon here. In other words, as my colleagues and I at the University of Washington have argued, clinical research translation is better understood as a translational cycle rather than a translational pathway, as I showed in the previous slide, in which basic science discoveries and developments must be understood and analyzed in the context of their iterative impacts on healthcare delivery and healthcare outcomes. So if we think about this in the context of clinical translational genomics research, socio-demographic characteristics that might and increasingly do, particularly in the United States context, but increasingly internationally as well, drive recruitment to genetic studies and a desire to diversify, because we need to, right? We've seen this data over and over again. We must diversify genomic discovery. But as we do this, this necessarily entails, for the reasons that Dr. Clark and others have discussed, the use of genetic ancestry information in the process of identifying replicable, robust gene disease associations. And then those associations that are ultimately defined by these genetic ancestry categories come back into a clinical encounter, especially in the United States context, but increasingly I think this gets complicated as we think about international programs of research, come back into the clinical encounter where patients are once more largely understood via socio-demographic characteristics, especially in a clinical context in the United States, and as Emer just showed us, in the context of health systems with respect to race or ethnicity. So these slippages not only happen in the context of the overall translational research cycle, but they can happen more upstream as well. So just take, for example, a major publication out of the NHLBI Top Med Consortium, a publication that I hasten to add, I am an author on. There's about 500 authors on this paper, but reporting initial findings from the first almost 54,000 top med whole genome sequences that were derived on the basis of 23 extant cohorts, which were brought into the top med initiative. A depiction of principal components for the data, color-coded by categories that were slapped onto the samples put here that are defined according to a mix of race, ancestry, or ethnic information. I'm reading directly from the figure label here, okay, provided by the study participants and or study investigators, right? So we have principal components of variation, we have complex population description defined by self-description or PI description, illustrates the rich complexity of the data, and yet later in the same paper, multiple subsequent analyses, and there are a bazillion analyses in this paper. If you haven't seen it, I highly recommend it. Multiple subsequent analyses report findings organized by those canonical large buckets of ancestry: African ancestry, East Asian ancestry, European ancestry. And then later, as shown in this depiction of estimates of effective population size based in patterns of identity by descent, by so-called population groups representing a mix of ancestry-based and US-based ethnic categories, for example, Hispanic, Latino. These slippages in this paper or function both of the need to distill all of that complexity on the left-hand side of the screen into simpler strata for the purposes of analysis, and by the fact that different analyses in this publication were conducted by different folks, who had different organizational schemas in their head. This is illustrating what's going on, and we see this happening over and over again. Part of the difficulty of managing descriptors in the Top Med Consortia was due to the inclusion of cohorts ascertained both within and outside of the United States. The same problem pertains for the Primed Consortium, which has only recently been established, and consortium leadership and the coordinating center, of which I'm a member, are currently considering how best to tackle population description in the context of the efforts of this consortium. Perhaps taking some lessons from the Top Med experience, perhaps learning from this committee. Unfortunately, I don't think you guys are going to finish your work soon enough to help us with this, because we have to get this organized and get the work done. And this is, I think, part of the problem in the context of many of these research consortia. But all of this complexity, notwithstanding all of the really great recommendations that have been made from people, it gets very much more complicated when the ways in which we are standardly thinking about ancestry are in this weird kind of munge of geopolitical kind of categories, such as some of the ones that Joanna described, but then we're also using racial ethnic categories that are distinctly North American and particularly American, and then we're trying to apply those in an international context. Whatever analytical approach is chosen within Prime is going to have tangible consequences, however, for clinical translation, as has become apparent in the recent work of yet another consortia that I'm involved in, the eMERGE Consortium. So eMERGE, which is in its fourth phase, is developing PRS, polygenic risk scores, for 11 different phenotypes that are going to be returned to 25,000 patients in 10 different healthcare settings, both adult and pediatrics. The PRS that are going to be returned were developed from pre-existing GWAS investigations conducted in collections of patients that were defined in a variety of ways. As a consequence, odds of disease risk have been estimated for and will be reported out with respect to different populations defined as shown here. This is some language taken from a FAQ that is going to be provided to research participants. Defined as shown here as reflecting Asian descent, African descent, European descent, and yes, once again, Hispanic Latino descent. What does descent actually mean in this context? That's not clear, and actually apparently varied from phenotype to phenotype as a function of the data that were available as the scores were being validated, and certainly is not explicitly explained in the test report that will be shared with patients and their clinicians. Now, this kind of blurring of the lines or kind of ambiguity around population categories may not inexorably lead to the kind of eugenic impulses that Rena Bliss referenced in her presentation focus on genomic applications this morning, but it is certainly set to sow confusion. Fortunately, we will be studying outcomes as part of this implementation project, and we may be able to say something about the impact of these practices on the use of this kind of information in clinical care.
You have 2 minutes, Malia.
Great, thanks so much, Rick. I know I'm finishing up. There are other examples I could discuss, so I'll just end on a cautionary and perhaps unduly pessimistic note. Now, the committee, as you know, has been convened at the behest of the NHGRI and other NIH institutes, and the NHGRI is, of course, on record as boldly predicting, perhaps because of the hope in the work of this committee, that research in human genomics will have moved beyond population descriptors based on historic social constructs such as race. But my boots on the ground experience in the context of these clinical translational research projects suggest, I fear, that this may very well be a pipe dream. And I hate to say that, but that's where I'm at right now. So I'm just being honest, okay? As I was recently reminded in contributing a manuscript to a forthcoming special issue of the Royal Society of the Proceedings of the Royal Society Part B, that's focused on the 50-year anniversary of Richard Lewontin's seminal publication on the apportionment of human diversity that Joseph Graves referred to this morning, no less a personage than Richard Lewontin demonstrated convincingly that the small proportion of genetic variation that distinguished groups historically understood as constituting human races was too small to warrant its use in biological investigation, and was of no social value, positively destructive of social and human relations, and therefore no justification could be continued for its continuance. And yet here we are, 50 years later, still having the same debate and the same discussion. This has suggested to me and my co-author, Jonathan Kaplan, that the genomics research community cannot ever hope to transcend the reality of social understandings of population difference, and that instead we must acknowledge the inevitable context dependence and fluidity of population categories and situate our genetic investigations alongside other, equally, if not more important, environmental contributions to disease risk. And we've heard about that in Dr. Mercer's presentation. We also heard about that in several other presentations in our session today. So in conclusion, clinical translation of genetic discovery is iterative, not linear. Genetics can inform but not transcend social understanding. Slippages in current projects illustrate the problem. And it's not clear that we can, or maybe even really that we should, move beyond social categories as we try to do our work carefully in the human genomic space. And just a big thank you to past and current collaborators in the context of these many research consortia in which I've had the pleasure to work. Thanks very much.
Thank you, Malia. Really good talk. And at this moment, I ask that my fellow committee members turn on your cameras. We will first take some questions from the committee, and then we'll open it up to the general public. So we had some really good talks. And do we have any questions from the committee? Okay. Well, I want to go back to the talk that Joanna gave, where she mentioned the geopolitical descriptors that have been used by 23andMe, and sort of how that evolved with input from the general public. And so my question is: so has there been sort of a place where you guys have fell in that space? Have you figured it out yet? I guess. You're on mute.
Yes, thanks. I would say that starting out just trying to be geographic, without so much of the political in there, but there's obviously always political in there when you're using a map. But we had challenges with people actually knowing what might be considered Northern European, even though geneticists are quite happy with that label. So we ended up having to go back to the country level labels. And that's what people are pretty good at, country level labels, even though those labels have changed over time. Pretty much around the world, people, can tie their, themselves to different countries in terms of ancestry. So I do think that's kind of where we ended up. And you know what, even more recently, we've been able to use that Google location thing where you just start typing, like I can start typing Utoxeter and it pulls up where my grandmother was born, in England somewhere. So you just have to start typing the name of almost like a village or town. And so people can get very precise with geographic locations with that kind of prompt and help from the tool that kind of takes you to a particular location. And so we've gotten very, very granular with location in recent times. And I think it's not a grid, and we don't have an even coverage across the grid, but it's getting closer to that. And that's pretty exciting.
So I guess the question would be, would you group individuals who are geographically close to those points? Would that be sort of how you would define these populations?
Yes. So you can do it at all levels. You can do it, we tend to do it like, down to like a town level, state level. And even, if we do maps, we're just doing like longitude, latitude. And you can do like, like put frequencies on a map. And so you're not really having to do much labeling. You obviously the map has labels in order to be interpreted, but you can get away from, the kind of level of granularity by having, like say a map with a gradient on it. That is pretty cool. And that's kind of getting somewhere that I'm happy with.
Mm-hmm. Okay, thanks. Sarah Titchkoff, she has a question.
Yeah, my question sort of expands upon this current discussion, and it pertains to Joanna and Emer's talk, because Joanna, you talked about this classification of people who have Indigenous American, Native American ancestry, and I was trying to figure out how you constructed that map and those labels. And if people are being ascribed to them based purely on genetic clustering, because I looked up your blog and it looked like there was some overlap between, some of these classifications in terms of geography. And then in Emer's talk, you showed, I think, a principal component analysis and clustering based on genetic data, but then on top of that was sort of self-identified ethnicity. And you would have like Finnish and Kenyan, but it looked like there was a lot of overlap with the African ancestry cluster and the European ancestry cluster. And how well could you actually distinguish people based just on genetics? I'm trying to understand that better.
Well, I can answer the second part of that question very quickly, Joanna, if you want me to, and then I'll pass it back over to you. So, Sarah, the plot that I showed, actually, it was a IBD clustering plot. Okay. So in 2 dimensions, actually, distance isn't a way to interpret it. But that's my bad. I didn't explain that very well. But they're actually quite, in multidimensional space, they're quite distinct IBD community clusters that pull out based on sharing within versus sharing outside those communities.
So you could distinguish, for example, Finnish or someone from Kenya.
Yeah, in New York, we can distinguish, for example, somebody with recent ancestry or born in the Dominican Republic versus somebody with recent ancestry or born in Puerto Rico with a greater than 95% positive predictive value.
Okay, thanks.
Should I go ahead and respond to Sarah? Sarah, you were asking about the overlap of those clusters.
Yeah, how they were determined.
So you have data from individuals who have, what's previously been identified as, Indigenous American genetic ancestry, and then we this was done by Kasia Britt, and then you look at the clustering of individuals. This is all genetic clusters. And then those clusters were kind of put on the map based on that geographic information from the participants. And yes, some of them end up overlapping. The clusters overlap geographically, but that means there's sort of genetic layers over the map. It was kind of, this was through guidance from members of the Indigenous American scientific community, and that's how we ended up with that set of labels.
Okay.
So we could have labeled them different things. You could have had, like, instead of a South Central, it could have been like Floridian or something. But we basically stuck with fairly.
But there were discrete boundaries, or you could actually distinguish someone from Great Lakes versus one of the others, or it was just like you said, you put a map of like different, I don't know, frequencies.
Well, the map is overlapping, so you can, but you do find that people have connections to one of those groups. Sometimes it's to two, multiple groups. Not everyone who's from a particular area has connections to everything that overlaps that area. So yeah, the genetic cluster, the whole thing is based on the genetic clusters that only have the genetic data in them. And then once you have those clusters, you find the labels based on the geography. And that's what the customer sees in their results. It took a long time to get there.
Okay, thank you.
All right, next question, Aravinda.
Yeah, first, thank you to our 5 speakers. This is, I think, each of you have been really quite thoughtful in laying out your perspectives, and starting with, the end, Malia. I don't know that we really even, my personal opinion, I don't know that we need to ignore the social categories, because obviously they're a very important part of at least the disease burden, which I'm sure is a very big motivation for the NIH and NHGRI. I wanted to just bring up an issue that I think, if you can work in your answers, or you can write to us after this, is one of the things I see is this overall reliance in today's discussion on GWAS studies. I understand, I'm very familiar with GWAS studies. I've done many themselves. In fact, in one of the, I think it was the second study published. So, but I would want each of you to think of genetic studies in a somewhat broader way. As we get the sequence, there are many other things we can do. And I don't know that we will serve ourselves as a community by being fixed only on GWAS studies. There may be other ways in which we will get to the basis of understanding these diseases, in which case there would be very different meaning. And this is really a plea to all of you as experts who can inform this committee, whether you do it today or tomorrow or write to us. The second is, I'm going to bring up this historical thing. It would be useful, all of us, to think that as the amount of data on genomes at whatever resolution increases, and it is increasing at a tremendously large rate. I'm not going to ask Joanna how many people have taken 23andMe, but I'm sure it's in over 10 million by my guess. That if it is, there will be more and more data, as well as with other studies. So the resolution with which we can get genetic clusters for the purpose of prediction may be very different than what we can do. I'll tell you just 1 minute on, because it's come up many times today, starting with Pilar, on doing grid sampling. I think if I look around, I've been around in genetics probably longer than most of you, but the idea first arose that I'm aware in a discussion I had with the late Alan Wilson. And of course, it was impossible 40 years ago to be able to even contemplate doing this, but the idea actually has been around. There are many ways of sampling species and individuals, and there's transect sampling. There are many ways of sampling. And I think in a way, it would be good if geneticists thought of other ways of trying to assess variation rather than only as populations. Populations are one feature. That sort of brings me to my actual sort of question, which is, in the genetic clusters, whether Joanna, it's yours, or Emer, whether it's yours, deriving these clusters depend also on other reference data. And so do you think, how do we get around that circularity? There's a little bit of circularity today, but how do we get around it? That we infer somebody's proximity to some population or region based on existing data, which itself is biased because we knew little when we started that study. I just mentioned them only as problems in our area. You may not have time to respond to all of this, but if you could respond to the committee, it would really help us in our tasks. But maybe Emer or Joanna, you can answer that last question briefly.
Well, Joanna, I don't know if you'd prefer to go first. Either way is fine. Maybe I'll start.
Start.
Oh, thanks. I don't think Joanna or I really had a lot of time to go under the hood a little bit in some of these methods. So just very briefly, and I don't want to speak for Joanna, but I do think that it's sort of similar on the 23andMe side. But interestingly, a lot of these methods, particularly the ones that are based on identity by descent, which is a measure of cryptic relatedness by shared long haplotype sharing, that tells you a lot about demographic events that occurred in the last 20 to 50 generations ago, so the last couple of thousand years of human history. Having reference panels and data in those assays can be very, very helpful to either seed them or to interpret them, but not always necessary. In the presence of, for example, strong founding events, you can often pick up, or specific admixture events, you can often pick up specific clusters, even if you don't necessarily have a reference panel or a population descriptor that you can apply. So, it is orthogonal information that can teach you about a lot of demographic events in the data, even if you can't necessarily interpret that with or apply a population descriptor. I don't know, Joanna, if you'd like to say anything more.
I don't have too much to add to that. I just think that the circularity, I feel like there is a way to get around it or get out of that circle a little bit if you're aware of it, by either getting more granular and just kind of checking on how is the circularity still there. I think there are ways to avoid really spinning around in that circle too far, and by basically going straight back to the IBD clustering, which Emer just mentioned, and that seems to me to be one of the ways it's working right now.
All right.
I want to add on the first question whether GWAS is the right way to do the way we do. So I feel including the epigenetic marks, the DNA methylation or other things, those are capturing more of the fluid environmental socio-environmental changes. As you know from population genetics, the SNP frequency requires probably generations to change, but those marks, epigenetic marks, could be a good surrogate for the socio-environmental factors what that will like. So it'd be good to integrate with other things. GWAS by itself, I don't think we pick the right thing, especially for this the racial disparities that may arise, and this is changing rapidly. So that couldn't be genetics alone. So, thank you.
Okay. Any other answers there? Okay, let's go to Anne. Anne has a question.
Sure, thank you. And again, thanks to all of you for your really insightful comments. I have a question for Dr. Mountain and then a question for Dr. Kenny, which I guess are probably both just points of clarification. But first of all, Dr. Mountain, it's interesting that you just recently in the Q&A spoke a little bit about using geographic data, even longitude and latitude. And you said, it's sort of getting towards, but it's not quite a grid system. And that made me want to ask you about something you said about grid sampling in your prepared comments. As I understand it, you said a grid approach is tricky because you have to identify a community to collect data from, if I understood. So I just wanted to ask you to clarify a little bit again, and I may have misunderstood what you said, but I wanted to understand that better. And then my question for Dr. Kenny has to do with the statement at the end of your presentation that I think you said might be somewhat controversial when you said that population labels were an important anchor to understand the gene environment impact on disease. when you say population labels, are you intending explicitly racial or ethnic labels, or are you taking a broader stance on what kinds of population labels might be an important anchor for such research?
I'm taking absolutely a much broader stance, and actually a much more agile stance on exactly what population labels, because different labels may apply depending on the question you're trying to ask.
Thank you for clarifying.
This question, and is quite a great one, because at one time I'm saying if you get down to longitude, latitude, you kind of get away from, you get closer to the grid, and yet with the grid approach, I'm just thinking of the way we go about obtaining samples from populations around the world, especially outside of the U.S. We have to work really closely with an ethics review board or a community advisory board in order to kind of get everyone comfortable with what we're doing, or even to introduce the idea. It's a little different in the US, where people have heard of 23andMe. They have a bit of a sense of what genetics is and what's happening. So I guess it sort of depends on if you're working within the US or not. Now, with the indigenous populations, we had a similar approach where we felt the need to, discuss the labeling with community members. And that's the grid kind of can work to a point, unless you have a need to work with any communities, in which case they may not fit on a grid very neatly.
Thank you both very much.
Okay, next question, Lance.
Yeah, let me add my thanks to all the speakers. Really informative and thoughtful talks, and I appreciate it very much. I have a question that I think any of the panelists could address, but it's really a reflection that was prompted by some comments from Dr. Mersha, Dr. Fullerton, and Dr. Kinney. And it really comes back, I think, to the core question, as we sort of set out at the beginning, about the idea of standardization and replicability and so forth. And at the beginning of the day, Dr. Osorio sort of suggested that we might be asking the wrong question and made an argument against standardization. I'd like to hear your reflections on that sort of provocation. Dr. Mersha, I thought I understood you to say that there was to be arguing in favor of harmonizing across different sort of systems, ways of categorizing human difference. Dr. Fullerton, I heard you say that we can't transcend the context dependencies, which seems consistent with Dr. Osorio's idea that we have to use categories in different ways depending on context. And Dr. Kinney, you just said in response to the last question that you were arguing for a sort of more agile way of thinking about the labels depending on not just context, but also the questions you ask. And part of what I'm taking away from that is the idea of some standard set of categories, some standard way of developing population descriptors may not be the right approach. So I'd love to hear your reflections on that.
Well, I can start the way I see it. So as we have this global consortia now, whether we like it or not, genetics is more global, and we're serving just one human population, one race. So, but locally, it's structured in many ways. Even in the country I grew up, there are different geographic regions, highland, lowland, all this different heterogeneity. So if we document that the way that, address the global structure and have a harmonization and ontologies like we do in clinical phenotyping, for example, we try to use similar description for clinical phenotype. And why don't we establish moving away from race and ethnicity, but having a common descriptor, how people, not just from scientist point of view. I was trying to address, we need to really discuss with the local lore and the local peoples and scientists in each country, in each region, so that we can come a consensus. potentially, if you develop a drug, you are trying to address reach out everywhere, not just for a certain group. So I feel that's a way to move forward, but I know it's not a straight line. But that's what I see the future of human genetics, at least to have a standardized approach like we do have in other fields.
I will jump in next, Emer, because I'm sure you have thoughts that are probably similar to mine. Yes, I think you characterize my views appropriately. They are very similar to Pilar's. And I think, and this is strong language, and so you feel free to take exception, but I think the error that we make in the genetics community is assuming that if we can come to some consensus about how to describe patterns of genetic similarities and differences amongst groups of people, that we can get to that kind of ontology that Dr. Mersha is speaking about. And it's possible that we can. I mean, I think that we are, as Aravinda noted, there's so many genomes that have been sequenced now. We are characterizing human genetic variation at an unprecedented scale. I think that we can come to some kind of defensible, whatever the word is, ontology, taxonomy, whatever, of human genetic variation. The difficulty, however, is that is necessary, but it is not sufficient for doing the work that we need to do to understand the role that genes play in health outcomes. And that's because, with the exception of those rare, highly penetrant variants that Emer specifically noted in her presentation, when we're thinking about polygenic risk scores, we are thinking about an entirely different class of variation where genes are not the only thing in play in defining risk. And as soon as we start bringing in non-genetic risk factors, the taxonomy or ontology that we develop using primarily genetic information will only be partially applicable. And so we will just not be able to, and this is people made reference to this in several presentations this morning, as well as in our session, about, a polygenic risk score developed in one population really appears to be only applicable to people from that very narrow population. But we keep wanting to expand out, because how on earth do we do that? That's enormously complicated. If you just look at the enormous genetic diversity in the biobank, we'd have to have polygenic risk scores for 15 or 20 different ancestral genetic groups. We can't do that, but maybe we have to. And so this is where I think it becomes really challenging, is if we're just thinking about genetic variation, then the kind of, and the harmonization efforts that Dr. Mersha is describing are absolutely essential to do this work, but they mislead us into believing it's all that we need to be paying attention to. And as soon as we bring in the other stuff, then it gets orders of magnitude more complicated, and that's where all this nuance that Emer was referring to comes in.
Can I ask you to clarify one thing? I mean, then why are we talking about groups and why not individuals?
Because Aravinda, that's how we make knowledge in genomic research. We associate genes in groups of people who have a disease, compare genes in groups of people who have a disease against people who are very much like them that do not have the disease. It's how we generate knowledge in genomics.
No, that's the beginning of the disease. But if you want to predict the effects, they have to be individuals. I mean,
so I think that you're arguing against the use of polygenic risk scores then. And that's fine. I don't like them either.
Well, I'd love to jump in there because I do think this is an important conversation, and I would actually argue against a dichotomy of a rare individual or family-based disease and a disease at a population level. I think as a community, we really ought to be thinking about a continuum of rare to common complex disease that has, to a greater or lesser degree, an environmental component. For some diseases that's going to be massive, for some diseases it's going to be small, but it probably still has an impact actually. And I think that the challenge here, and I alluded to this in my summation, is this almost a tension, is that we have to think about how we can accommodate both. And that's what I was referring to when I articulated that this needs to be agile to the question that you're asking. And I think the BRCA BRCA variants are a very good example, because that really is the example that encapsulates both. These are inherited, monogenic, large effect variants that we know are not necessarily fully penetrative through the person's life, and that there are both environmental and polygenic factors that play into their penetrance, issues of penetrance and expressivity. So, and that's the way, you know, the sort of the more integrated way in which we can think about these things. And that's the sort of a framework, I think, that might be helpful to think about those things. So what kind of solutions can we come up with that would accommodate both?
Okay, great discussion thus far. Lance, you don't have any other questions, do you? I see you put your hand down. Okay, great. Before I open it up for the general, the questions from the general public, I want to go back to the beginning of the discussion of the talks. And so Tess had mentioned the era and the issues around admixture and ancestry, the estimation of admixture. And I think Tess also brought up this idea that many use, including myself at times, that ancestry could be sort of this proxy for biology, this genetic background, this, you know, whatever that set of complex genotypes that are defined within that particular group are, you know, it's looked at as sort of like a biomarker. But it was also brought up by Tess that ancestry correlates with SES in ways that could be hidden in many cases, but in most cases it's pretty evident. And then I thought I heard it was either Emer or Malia mentioned that ancestry was a proxy for the environment or the social component. And so I'm a little confused sort of how I'm trying to get some utility here in terms of ancestry. So any comments on that? Okay, let's go back to say who said ancestry was a proxy for the environment, for the
Yes.
I'll put up my hand to start. I'll own up. Yeah, well, I think we're talking, so I think maybe part of the confusion is we're talking about two ways in which genomic, the genetic information is capturing components of social and environmental determinants of health. One is when we're explicitly using genetic information to infer something about our human history, and that itself is by cultural ties, by heritage ties, by political, social ties, is capturing something about the environment. But the other is when we use these type of aggregate scores where we're summing over a genome and we're constructing these tests like polygenic risk tests, what we're often including into those models are not only the genetic determinants that are associated with the outcomes of interest, but also the structural determinants, whether it's population structure, that are built into those scores as well. So they're capturing both, which is why as a field, standardly, what we do is we generate a polygenic risk score in a population, and then we look at the transferability or generalizability across populations. We often find it's less generalizable. It's both for genetic fact reasons, but also often for some of the most social determinant reasons that are less transferable.
Yeah, Eric, I was the one who said ancestry is a proxy for term. In estimating ancestry, we always determine admixture. We don't really directly measure ancestry. So because we don't know all our ancestors, we sometimes when we estimate admixture, we give, African American 10 generations from starting of admixture. In some cases, you see literature saying 8 generation. In some cases, 15 generation. So all those are based on some inference or simulation work. So because of that, the estimation, the admixture proportion is not really accurate at all to start with. Then when you interpret that to ancestry, that's again, all those errors, including the reference. Again, use the reference population, at least so far. Maybe as we get more sequence data, we may go away from reference. But now you need the reference population. For example, for African American, use African Yoruban and European. But there is also Native Americans within African American. We just discard those components. So the way we estimate as ancestry right now, to me, is more simplistic than accounting all diversity. So because of that, the estimate is not a true reflection. But sometimes we tend to say ancestry is a better reflection than race ethnicity. For me, both terms have their own limitation.
Okay.
And I don't think I said that, and I apologize if I did, but I would love to invite Melinda to weigh in, because it does seem to me that this way that we think about things, to what degree does the notion that an ancestral designation, however determined, could be a proxy for social determinants of health? Does that work in the UK setting? I mean, maybe it does. Probably
I guess there's a few things that I've been thinking about. One is I know that this is a group to focus on race and ethnicity and ancestry, but it's interesting to think about this transferability across sex, across age groups and the life course, and across the socioeconomic status. So this is a larger problem, and I think it becomes very apparent here, but I think it's a larger one. These are sort of similar discussions that we've been having in the UK, but you might have heard the horrific report that came out maybe a month or two ago, where the government announced that structural racism doesn't exist in the UK. So there you go. And it got a huge backlash, as you can imagine. So these discussions are really happening here. But I guess, my point comes up to, and I think Joe raised it in the last talk, is if we look at the explanation of your what's explained in terms of variance in your R squared, these polygenic risk scores, in terms of your explained variance, especially for complex phenotypes, remain very low. So, just throw in some of your education or your socioeconomic variables, and they'll explain health in a much more forceful way. So I guess I stand back and look at it and think, well, yeah, we can focus on this. And I'm on this board of Our Future Health, and there's a big focus on these polygenic risk scores. And you're thinking, yeah, okay, but we kind of know why we have structural inequalities in health. And you can intervene on them in a much more focused way. So those are just some of my thoughts. And I guess what I would like to see, and this is statistically impossible at the moment, but is a consolidated risk score, something that includes, we need these large samples for G by E and gene environment interaction, but I think we need to think in more complex ways. So yeah, I guess just sort of standing back, looking at it, just keeping them separated. They're both picking up multiple sort of traces of each other. The genetics is picking up traces of the environment and vice versa. I think we need to move beyond all of this, and statistically it's impossible at this moment, but get something more consolidated, I think.
So you're into consolidation. That's your position. I appreciated what you were saying about this, intersectionality and the multiple sort of types of diversity of data. And so I'm glad that you chimed in, because many of us think that this is a US situation, right? And it's unique because of the history of race and racism in the United States. But it's clear that it's, as other countries emerge from the cloak of honesty or whatever, or dishonesty, then we see that it's much more evident outside of the US also. But I also want to go back to the beginning when Joe talked about, this race and racism. And, I think part of what we are tasked to do here is obviously, there has to be a change in terms of how we define populations in genomic research, not just for the this social checkoff, but for the science, so that it's much more robust and future directions can really take advantage of this new thinking. But there's so much baggage with the history of race in America. So how do we, just the fact that, let's just be honest here, when we talk about ancestry correlating with SES, that's because of racism in America. That's because ancestry correlates with skin color, right? Skin color is a social strata for opportunities and access. And so, that how do we sort of disentangle, that history? Any comments?
One of the figure probably was quick to go through. We did the mediation analysis. So that's where the ancestry was highly correlated with hardship, SS, all these things. But ancestry wasn't directly related to asthma. Mm-hmm. But it was mediated through other factors. So the higher hierarchical ancestry is correlated to hardship and other factors, and that hardship is correlated to asthma. So there is this kind of intermediary effect. Mm-hmm. Although genes directly not affecting asthma, but your genes obviously important. It goes through other factors depending on your percent of ancestry. So that kind of dissecting the relationship could give us some kind of clue.
Yeah, that's a great point. I think back at what Melinda presented, where she showed the different polygenic risk scores, and then the strata of SES, within each one of those risk scores, that's really telling, because maybe those risk scores do work, but for a particular strata of SES. And I think we really have to integrate that a lot more in these sorts of studies.
Yeah, and I think you can do it for type 1 or type 2 diabetes. You can do it for cardiovascular disease. There's other papers that have done that, and just shown this. And it's related, to multiple things. It's related to the samples. But, I just think it's really important to stand back when we're looking at all of these things. And what are we picking up in these scores as well, too? And that's something, just to stand back and reflect on. And we're talking about, you have to do homogeneous population to homogeneous population. That's very difficult to do. But also just acknowledge that some of the things you're picking up in the polygenic risk scores aren't all genetic. So I think that's, something that, I don't know, maybe I'm sure others have opinions on that, but looking at it from a social science perspective, when you see these polygenic scores predicting radically differently, and they're very stratified by socioeconomic traits. It seems quite clear.
Yeah, I'll just completely agree. And I think the disentangling part is the very important part. You can't just jump to think that that's because there's a genetic impact. In fact, there could be no genetic impact. The correlation could be explained completely by non-genetic factors. And so we do, but at the same time, that's why I use the word anchor. It does point you to where you have to look. And I see that as the first step.
Yeah, I agree with that. But I think it leaves me with a question, and this is something that I worry about a lot, is that let's say we can do this work really, really well in the biobank, for example, and really disentangle the relationship between genetic ancestry and sociocultural descriptors, social determinants of health, measures of healthcare disparity, for example. Is what you learn in that healthcare context, then, and this gets back, this is what Melinda's talking about. Is it translatable elsewhere in the United States? Right now, we seem to be hoping that it will be, but I think that's an open question. It's one that concerns me as we debate these issues of how to, the concern about labels misrepresents the deeper process level questions about how we are understanding the role of genes and other things in health risks, and how what we learn in one context can fruitfully be taken up and used in a different context, either both within the United States, which is remarkably diverse, right? And then internationally. I just think it concerns me.
So, Aravinda, last question from the committee before we open up to the public.
Yeah, I think all of you have sort of made this last point that Malia, you sort of just brought out very, very cleanly. I think many genetic studies, particularly GWAS studies, have no measures of the environment or anything else. And even when they do, well, the first thing the geneticist does is to regress them out. Sex also covers a lot. That's the first thing, we regress out. And even though there are many traits in which, just even measured sex, just of the number of X chromosomes, explain about 30% of the variation, much more than the other genes do. So I think what you're pointing out, one way to look at the problem, is that perhaps we shouldn't be doing all manner of studies on all genes and all phenotypes. That just the structure of what these studies say, that is, they could be exemplars where you do the same kind of studies in multiple locations in a similar way in multiple parts of the world in some designed way that we could learn. I'm just wondering whether I should make that inference. Just the last thing that Malia, you brought up, Melinda, you brought up, and Emer brought up and tested that perhaps we should learn how much, even for a few traits, genes matter relative to other things that we could measure.
Yes.
Completely agree. Yeah, I think it's a very important summary.
All right, so I'm going to take a question from the public here. This is, I'm horrible pronouncing names, so maybe I shouldn't attempt this, but this is a Bamidele Iwa Locum says his question is, I am from Africa and I say that genetic genomic studies are still in the infancy in the continent, despite the enormous genetic diversity in the continent. Are there information on ethnic descriptors done in Africa, especially in countries like Nigeria, where over 30 ethnic groups exist? Any takers on that?
I mean, I'd certainly jump in and point to the work that the H3 Africa Consortium has done, and some really wonderful ethno-ethnography of different groups across at least the cohorts that are in that study, which is does not include, obviously, it's much bigger, much bigger or smaller than the continent of Africa, but it is some really nicely exquisite work to map that out that has been done.
Right. There's a comment in the chat from Sarah. It says actually there's 400 ethnic groups in Nigeria.
I was just going to say Sarah should have actually been the person to answer that.
Well, I can just address a little bit, which is that even with the amazing work H3 Africa has done, there is this confounding still of labeling groups by geography. Kenya, Uganda, and then very rarely, some groups will do by self-identified ethnicity. Others will not. So there's a lot of variation, but not quite as much on self-identified ethnicity. It's usually by geographic region.
Okay. So question by Carrie S. This is aimed mostly at Dr. Kenny or Dr. Mountain, but anyone who wants to answer can. Would ancestral native language be a slightly more accurate way to differentiate similar groups that might otherwise be lost into a bigger group rather than where their grandparents were born? So use language as a grouping.
Rick, I'd like to jump in on that question. It is, we have found, looking at language linguistic groups across Atlantic Africa, we've been able to distinguish clusters that correspond to those linguistic groups, which was quite powerful. You know, I wasn't sure it would work, but it did. So yes, the language information has been very powerful, and I would say more powerful than the precise geographic location and so on. Yeah.
Do you think that would be a more difficult sort of descriptor to manage than geography?
I'm not sure. When we use the ethnologue, in many cases, when we don't have, other information, so I don't think it's that hard to manage that linguistic information. Plenty of people have studied linguistic variation across the entire world. So there's some databases, but I like to look at it, bring it all together, and just, you're going to slice the data in all the different ways and see, what maps to the genetic clustering and get some idea of what led to that clustering. I mean, language can be a barrier to mixing, and as can geography. So language can be part of the population history, and that's why it can be valuable, but it's not the only factor.
Okay. Question by Carolina Mays. This is a PRS question. Given that many individuals in the US and around the world do not fit within a single ancestry group, what are the challenges for translational genomics if utilizing population descriptors is unavoidable? To what extent do technologies like PRS pin individuals to a particular population? Anybody?
I'm going to jump in, but I probably am the least well qualified. But I think it is a problem. Now, of course, we can do increasingly sophisticated things as we look at patterns of genetic variation within and between populations. For example, and I think Emer talked about this. I believe Andy did this morning. We can estimate ancestry locally and in particular places in the chromosome, right? And perhaps, and some people think about this as sort of transcending this concern about taking a person from a particular region of the world who probably very likely has mixed genetic heritage, and not have to put them in a single box. So we'll just put each of their SNPs into a single box. And that may solve some of this problem. And I do think that's how people are thinking about making sense weighting of the information that Andy referred to this morning in the context of polygenic risk estimation. This does not take away from my other concern, which is once we have a way to weight the SNPs in the polygenic risk score, we are thinking about associations between those genotypes with phenotypes that are measured in populations. We cannot do that same aggregating down to a chromosomal level with phenotypes, I think. But Emer or others, jump in and tell me that I'm getting this wrong, because I would love to be corrected.
Well, I certainly agree with you to the extent that I think that the closer we come to actually directly modeling demographic forces in populations in these models that we use, that we're aggregating and actually capturing that information, whether you want to call it genetic ancestry or whether you want to call it admixture or whether you want to call it demography, we can argue about that, but whether or not you're modeling that becomes very important. So if you're ignoring that, or you're trying to, as Aravinda pointed out, I think we do a lot in our field is we adjust it out, we confound it out, we treat it as a nuisance factor. That's where you're going to run into problems. You actually have to grab the bull by the horn, and you have to explicitly model the population you're interested to produce a tool that's going to predict very well. And that's how you have to actually build out your models. I don't think that it's only going to be statistical solutions, but I do think we need to get a lot more sophisticated and nuanced with our statistical solutions.
All right, any other comments?
I just wanted to add that there may be things you can do that don't solve the problem completely. For instance, if you know something about, somebody's admixture proportions, maybe you say, well, a PRS based on this part of your ancestry would predict this, and then on the other part would predict this, and you're somewhere in the middle, maybe. I mean, there could be some kind of confounding, but there may be something you could, maybe you just get a bigger variance on the PRS score or something that, I don't know. I just think you're not, that is one of the hardest things when you have, when you want to do something personalized and that person is a one of a kind. There's no comparison set.
But I think with increasing immigration and just seeing these populations, we have the census coming out here, that increasingly these mixed groups, if you look at the migration history. So, I mean, it's something to be reckoned with. It's seen as sort of a statistical problem, but it's a reality, all these admixture and these mixed populations.
Yep. Aravinda, you have a question?
I just wanted, some of the speakers to clarify. Joanna, I get your point, because that's sort of generally the way I think many people, including myself, think. I guess, Malia, I'm still trying to understand your conundrum. Your conundrum is that we can do all of that. We can find SNPs, maybe even factor specific, some other risk factor specific genes, anything else. But then the connection of a genome's worth of this to phenotypes is not really measured in any singular population.
Yeah, I guess so. My question, Aravinda, is, and I don't think we have an answer to this yet.
Correct.
It's like, let's say and people are very interested in, and I know there are people right here in this room who are doing this good work, of developing polygenic risk scores, which will be trans-ancestral, right? That they will not work just in a single population, but will work across populations. And I believe we know enough about the way that genes vary within and between populations that we can do that work well. There is a set of SNPs that we can identify for particular phenotypes, which will get us there. We then need to take the distribution of scores for a cohort and associate that with disease outcomes. And it's that second piece of the exercise that is necessarily always has to be done in this very population-specific context, so that if I develop a polygenic risk score which predicts risk really well in folks of Chinese ancestry who live in New York City, will that work for people living in mainland China or not? I don't think we know the answer to that question yet. I think we have some assumptions, but I agree with Emer that we need to be doing a much better job with our modeling.
It is a testable hypothesis.
It is a testable hypothesis, but it's one that has not been tested yet, as far as I'm aware.
Okay, all right. So we're going to go back to the questions. These are some really good questions that we're getting for this session from the public. So Anna Lewis asked, and this probably can go to Tess or to Emer, if one is jointly statistically modeling race and genetic ancestry, to what extent does one have to worry about collinearity? And if we do need to worry, do we need any new methods?
Tess, would you like to go first?
Well, it's a very new thinking. I never thought jointly modeling ancestry in race, but I think it's this too. Most study shows at least global ancestry is correlated with socio-environmental factors, but that correlation may not be in a causal way. So adding in the model race, as the person mentioned, it increases the collinearity because you are becoming more circular, including the similar factors. So I don't think it can improve in my view, but that's worth trying, and maybe don't know, maybe you can add something?
Yeah, certainly. I think there's a few methods out there and a few groups working on these problems. And the question is, I think, to the crux of what Anna is asking is what's the interaction term between the genetic ancestry component and the genetic component, or the environment component, etc. The answer is there is some collinearity, but it's not always linear, that there can be other types of interaction between these things. And you really that's what I'm saying. I keep pushing. We have to build these models and test these hypotheses, because the answer isn't always going to be intuitive. But I don't see why we can't build these things in, because we do consider many other different types of factors and covariates in our models.
Right, right. And there's no reason to assume that it's going to be the same across those groups, right? I mean, even within, let's say, African Americans in particular regions, they could change. I mean, I see from data that Ancestry correlates with SES in certain settings, certain communities, but not all. And so local history matters, local experiences matter. That context is important. So you can't assume that it's going to be the same across.
Yeah, exactly. Or across phenotypes.
Right. All right, next question. And we have about 5 minutes remaining for Q&A. This is from Barbara Koenig. There has been very sophisticated discussion on the social category of race and its history, but very little discussion of where the idea of ethnicity comes from. How is the committee planning to flesh this out, this issue of ethnicity? And is it important in this discussion? I added that piece. Any takers? Because, so often we kind of dismiss ethnicity, right? We use it to differentiate Hispanic Latinos from whites, right?
Yeah. Sorry, go ahead.
I just want to quickly say that, for 23andMe, the term ethnicity is used fairly loosely and acknowledged as such, that it goes far beyond Hispanic, non-Hispanic. It's more about a cultural affiliation and social affiliation. And it, just, so I don't know, it's part of the term, I'm sure the use of the term will continue to evolve, but definitely haven't tried to even define it. Yeah. Tess?
Yeah, I think I also agree what Joanna said. The more I understand race ethnicity, actually ethnicity becomes more relevant for genomics because it captures really, it's not political, of course there's a political, but race is just black or white. It's kind of hierarchy, but ethnicity, it can be different in different racial groups, but it's more, your affiliation, your lifestyle, all these commonalities. So that may reflect more genetic differences than race in my view. So I think that's a very good term to look at even further than what we right now.
Charmaine, you have your hand raised?
Yes, I would just like to, I'll just start by thanking all the speakers for such stimulating presentations. But to respond to Barbara's question about whether the committee is going to look at ethnicity, surely ethnicity is part of our charge, right? Our charge is to look at the use of race, ethnicity, and ancestry. As Aravinda said before, we haven't started writing a report yet. And so nothing is set in stone in terms of how we're going to approach the report. And as the committee gets back together and culls all the information from today and begins working on the report, then we'll figure out to what extent and how much we delve into each of these labels. But it's definitely part of our charge, and we will be looking at ethnicity as well. I mean, it's part of the conversation, and it's used as well in many ways in these studies. So yes, how we'll figure it out as we go along.
All right, thanks. I believe we have a minute or so before break. So any final comments or questions? quickly go to before the break? Okay. Well, if not, then I want to thank the speakers for this great session, and we'll now take a break and reconvene at 4 p.m. Eastern time, which is 1 p.m. for me.